Hello everyone. Today we will discuss about the Bacillus. Here are some general characteristics of Bacillus. Basically, they are gram-positive bacteria and they are rod-like in structure. They form spores to reproduce asexually. They are obligate aerobes except anthracis. There are 266 named species of Bacillus. So here we will discuss about two types of the Bacillus. Number one, Bacillus anthracis, and the second one is Bacillus cereus. Now, the Bacillus anthracis. They are causative agent of important zoonotic disease, the anthrax. They are large rectangular rods arranged in chains, bamboo stick appearance. They are non-motile and they are capsulated and they are facultative anaerobes. Now, the importance of Bacillus. First, pathogenic bacterium seen under microscope. First, pathogenic bacteria isolated in pure culture, and the Bacillus is the largest pathogenic bacterium. So let's talk about its epidemiology. The animal anthrax and the human anthrax. First, the animal anthrax, primarily a disease of herbivorous animals. Spores remain viable in the soil for decades. And the human anthrax, from agricultural exposure to animals, infected animal products such as hides, hairs, bristles, and wool. So here are some clinical manifestations. Animal anthrax is basically a zoonotic disease. More commonly, herbivorous animals affected by this by ingestion of spores presented as fetal septicemia. Infected animals discharge large number of bacilli from mouth, nose, and rectum. These remain in sole source of infection. So let's talk about the human anthrax. Spores of Bacillus anthracis are absorbed by skin, or you can say cutaneously, or by inhalation of spores, or ingestion of carcasses of animals dying of anthrax, or through termites, rarely. Now here are some clinical types: cutaneous anthrax, pulmonary anthrax, intestinal anthrax. So first of all, cutaneous anthrax. So what happens when you get infected by cutaneous anthrax? So here are some symptoms. A group of small blisters or bumps that may itch. Swelling can occur around the sore. A painless skin sore known as ulcer with a black center that appears after the small blisters or bumps. Most often, the sore will be on the face, neck, arms, or hand. Now the second one, intestinal anthrax. So symptoms of intestinal anthrax are fever and chills, swelling of neck or neck glands, sore throat, painful swallowing, nausea and vomiting, especially bloody vomiting, diarrhea or bloody diarrhea, stomach pain, swelling of abdomen or stomach. After the intestinal anthrax, now here is pulmonary anthrax. The symptoms of pulmonary anthrax are similar to intestinal anthrax. So here is fever and chills, chest discomfort, shortness of breath, confusion or dizziness, cough, nausea, vomiting or stomach pains, headache, sweats, extreme tiredness and body aches. Let's talk about culture characteristics of Bacillus anthracis. Anthrax bacilli are aerobes and facultative anaerobes. The optimum temperature for its growth is 37 Celsius, but growth occurs over a wide temperature range of 12 to 45 Celsius. Optimum pH for its growth is 7 to 7.4. They can grow on ordinary media. On nutrient agar, the organisms produce large raised opaque grayish white colonies 2 to 3 mm in diameter with an irregular fringe-like edge. Under low power of the microscope, the edge of the colony is found to be composed of long, interlacing chains of bacilli. Resembling curled hair lobe, this gives them the medusa head appearance. Virulent capsulated strains from rough colonies, while avirulent strains from the smooth colonies. Colonies on blood agar produce very slight hemolysis. A selective medium known as PLET medium, consisting of heart infusion agar with polymyxin, lysosomes, ethylene diamine tetraacetic acid. Thallus acetate has been devised to isolate B. anthracis from mixtures containing other spore-bearing bacilli. In a gelatin step culture, there is growth down the step line with lateral spikes that are longest near the surface, giving inverted fire tree appearance, with slow-like friction commencing from the top. When B. anthracis is grown on the surface of solid medium containing 0.05 to 0.5 units of penicillin per ml, in 3 to 6 hours the cells become large, spherical and occurs in chains on the surface of agar, resembling a string of pearls. This string of pearls reaction clearly differentiates B. anthracis from B. cereus and other aerobic spore bearers. B. anthracis is susceptible to gammaphage. This is another useful test which differentiates it from the B. cereus. The diseases that we have discussed are treated by the following antibiotics. So here is another type of the Bacillus anthracis which is very important to be discussed. The Bacillus cereus. Cereus is a Latin word which means wax. Bacillus cereus is a gram-positive bacteria. It is a rod-shaped in structure. It is a facultative anaerobic and it is a spore-forming bacterium. Commonly found in soil, food and marine sponges. The toxins that Bacillus cereus produce are emetic toxins. It causes vomiting and abdominal cramps within 1 to 6 hours. There are some foods that can cause this illness include cheese as well as starchy foods such as pasta, pastry and potatoes. After the emetic toxins, we will discuss enterotoxins, diarrheal syndrome. Ingestion of the bacteria causes production of toxin in your small intestine. You usually get food poisoning 6 to 15 hours after eating contaminated food. Foods that can cause this illness include fish, dairy, meat, sauces, soups, and stews, vegetables. Now, the non-intestinal illness. Symptoms vary based on the type of illness. Endophthalmitis causes the highest amount of the severe illness. Symptoms of this life-threatening eye infection may include eye pain, fatigue, fever, high white blood cell amount, leukocytosis, low vision, red eye, ring-shaped corneal ulcer. Infections that are caused by the Bacillus cereus are treated by the following antibiotics. But here is one thing which should be noted is they are resistant to penicillin. So that's all. Thanks for listening.